now today I'm going to focus on the question, what if a method exists, what can we do with it? And one issue is disease data, another one is something like what I call beyond singles. Let's begin with disease data. So the first question is, or the first story is, we are going to simply apply 3 different methods here: SNAP, machine learning based, polyphen and SIFT, on a set of disease variants, namely OMIM, online mammalian inheritance in man. So these are an individual SAF, the kinds of situations where an individual SAF, an individual amino acid change, causes a disease. Every single one of them. We run this data set and we simply ask what fraction of those is predicted to have an effect by one of these 3 methods. You see these 3 results. What do you find? Which method is best? Or what considerations do you have before you answer that question? So first we have to understand the data, completely understand that. So on the next slide, what I'm going to show, what I show here is the training effect. So this is cross-validation. And training neutral. I call it training because this is part of the cross-validation set. This is not part of the cross-validation set. So what we do see here is that for a part where the effects are in the cross-validation set, this is still higher. So the set of OMIM predicted by SNAP is more than, in fact, for its same level of what it was trained on. Again, this is the cross-validated result, but it's the data it saw for development. And for the data it saw for development, effect still has a lower, it predicts more effect in the OMIM data set that it has never seen than in the set it was training with the positives, okay? So this number here for OMIM is higher than that number. So clearly, part of the reason why OMIM is predicted better, is stronger, is not because the system over-predicts all the time, okay? Then it would have this signal here higher. That's the point, right? There's a difference here. And then you have to do something like that for all the others, and you sort of, step by step, you argue that in fact there's a variety of steps that show that this is sort of more likely to not really be a forte of the method, but something that has to do with training. Now, I'm looking at 2 different data sets. They are very differently sized. OMIM at this point here is 5,600 roughly. This OMIA. OMIA is the same as OMIM in animals. Online mammalian inheritance in animals. This is where it comes from. It's only 117. It's more difficult to get those out there. Hand annotated, yes?
Sorry for this naive question, but what we are trying to predict, is it if a mutation is going to cause a disease, or what is it?
No, it's not a naive question. In fact, the problem is that we don't even have a clear answer to your question. What we are training on is molecular function. Most of the data points we have is the effect of a variant upon a function of a protein. Okay? So therefore I would argue we tend to in fact try to predict what the function of the effect of upon function of a protein is. What we really want to know at the end of the day, if we compare the variance between you and your neighbor, is how will that help us to sort of treat you differently? And that is more in the direction of disease. So we somehow assume that there's a connection between these 2. We somehow try to get to the point where we get from differences in genotype to the level of differences in the sensitivity to disease.
But do you train with data of healthy, like,
yes. So this is this.
Gene and then predict the function, or
now we get into
genes with point mutations of known diseases.
So let's stick on these numbers here. So there now is a bunch of methods out there that train only on human variants in human. Those methods mostly are biased by, or not biased, mostly use essentially OMIM. Because 5,600 measurements for human proteins you actually don't have. So when these methods are developed, they're mostly trained, at least on the level of effect, they're mostly trained on disease, on variants that have an effect on a disease. Yeah?
So do you feed in many different variants, but just look at one gene, like many different versions
yes.
of this one gene? Okay.
It's only one gene. Again, so all of these methods, essentially the idea, I don't have a pen. The idea is you say you have 5 consecutive residues, and you take out what's the effect on the central one if you mutate that central one?
Okay, but like the whole training set is about one gene as opposed to the
Yes.
whole genome. Okay.
Yes. So there's no method out there on that level that takes an entire set of proteins, if that's what you're asking. Yes?
Again, the main problem is that, if I recall correctly, we have those trigger variants which we actually denote as effect, as having an effect, right? When training data, we have the spectrostatic and the trigger variants.
I'm not entirely sure what you mean by trigger. You mean trigger the SAFs for which you have an experimental annotation?
Exactly. Yeah.
Yes.
Yeah.
I'll call them trigger now. Okay. Yes.
No, trigger, because you made two distinctions. Either a trigger SAF or a retrostatic SAF. So
rheostat and toggle.
Toggle, not trigger. I'm sorry. Toggle.
I'm sorry that I didn't understand what you mean. Yes.
Toggle.
Yes.
So we mainly train effect based on toggles we observe, right?
We assume that the answer is right. So we assume that the answer to your question is yes. But in terms of the way we get the data, effectively we get the ones with a stronger, So the more subtle it is, the more difficult it is to measure.
Right.
So since most of what we have was more easy to measure, we assume that most of them would be the toggle type.
But especially for
therefore.
diseases, the rheostatic changes can have a dramatic effect.
Yes, but these in fact are 5661 where it's toggle. These are, you carry that variant, disease, full stop.
But
that's a clear case.
Due to a chain of rheostatic.
They always start with a thing. You take the same cell, you introduce one SAF,
right.
You have the disease.
Right, but as you are...
There are downstream effects. But they all start with this SAF. Yes?
Right.
When training, do you also take in structural information? Or
No, so the answer is wrong. I said no, and that is a wrong answer. Some methods do. The method that I, for instance, look at here, SNAP does not. It takes predicted secondary structure, predicted solvent accessibility, it takes predicted contacts, but it does not take actual structure. So, to repeat, this is disease in people, and this are the type of variants we see here, but diseases in animals. The reason why they are so small, why the data set is so small, is because they are less well studied, because it is more interesting to study diseases in humans. We care more about ourselves than about animals, as simple as that. And the data sets that exist that came much later, and therefore also smaller, are also more difficult to parse, interestingly enough. Yes?
Is it then also non-redundant with man? So are
yes.
they non-existent?
Yes.
Because I mean,
I do not believe.
Cell diseases could be common through animals.
Oh, there's a funny twist to that story. So the funny the twist to that story is this: what I keep arguing about, the E7 to V or something like that in sickle cell anemia. SNAP cannot pick it because in many animals this is actually observed, the variant.
Okay.
So SNAP is totally fooled because it looks like something that in evolution works. Because rats can live with it. A lot of animals can actually live with it. This is very specific to human, yeah?
I have actually two more questions.
Yeah?
What's the gene we're looking at in this plot?
In this plot, so let me put that differently. We are looking, I cannot quite remember the number. I believe we are looking at something in the ballpark of 2,000 different genes here. In these 2,000 different genes, we have 5,661 SAFs. So individual amino acid changes.
Are you looking like at the ADH or at a certain enzyme, or what is?
Those are all kinds of proteins, yes. They're not only certain enzymes. So these are in human for all the type of variants we are talking about. So if you have a variant on the gene that immediately leads to a disease, this is typically called the rare diseases. This is the entire set of rare diseases that are currently described in the literature on the level of the protein. So this involves about 2,000 proteins, and there's essentially anything you can name, from structural proteins to enzymes. You have transcription factors, you have almost anything that you can name in this set. It's 2,000, so it's a subset of 20,000. What we do not know is whether every protein ultimately will have one of these rare diseases. Maybe, we don't know. This is where the research stands at the moment. And this has has evolved dramatically in the sense that 10 years ago this was less than 2,000. So it has, well, a factor of 3 in 10 years or something, yes?
So on average, every protein has 3 indexes, or
so you have to be careful. So I do not know, unfortunately cannot answer your question. What I'm saying is we have 5,661 salves. They could actually hit on the same residue. Right? It could be that in one patient this is E to V and another one that's an E to L. That would still count 5,000. So 5,661 is 5,661 people who have a unique variant that is in a sense of causative relation related to disease. That's what we have. For animals, the same number is 117. And again, most likely these proteins are not in the same. I don't believe we ever looked at the overlap of the families or something like that. And I cannot remember how many proteins we have. I believe this is fairly unique in this sense. I believe we have very few that are double here. But my question to you is, and the error estimates are such that, let me just assume you see the error. It's not true, but assume I stated you see the error estimate. So it's so small that you don't see it, right? That the error bars are falling within the line. What would you then argue that you see? These are predicted effect of 5,600, predicted effect, so, yeah?
But the ones that we've annotated in animals are like those kind of super basic schemas that they're easy to learn.
Yes, you go 3 steps ahead. So let's put the first step down. This bar is higher than that bar. Which means that it looks as if I can trust my prediction method, effects in animals have a higher, variance in animals have a higher effect, tend to have a stronger effect than in humans.
Stronger is easier to see.
And you say it's easier to see. That is a very, very interesting explanation.
It has a higher effect if you say...
On average it does. Oh, there's something else. In Tuesday I went through too many slides too quickly. But one slide said that there is a correlation between the strength of a prediction and the size of the effect. So stronger effects are predicted more strongly. So if you have a higher ratio, that tends to indicate that on average the effect is stronger for that set. Okay? So this would then be a little bit stronger. And ultimately one explanation is that you have, very interesting one, animals just have the ones that are easier to predict. Another one would be, actually they are stronger because what we call disease in human is something else than what we call disease in an animal. So if I have a big bald spot on my head, I call that a disease because I'm unhappy. The cow is not going to be unhappy. So cows don't go to the doctor for having an empty spot. Ultimately, that may be part of the explanation. When we look at this set, so in this set is so small that we can look at it, those are all dreadful diseases. Here you have many dreadful diseases, but you have things such as no hair in there. And you know, again, if you don't have, I'm not saying it's great to have not hair, but it's not as dreadful as sort of to die. Well, anyway, let's get over the ethic of that. Anyway, so this is interesting that you can, with a prediction method, see some trend in a data set that appears to have some reason behind it. Now here's another thing that we found interesting. So we take this human variant and we put it into a mouse protein. And we insert in this mouse protein the variant and ask how much is the effect on the mouse protein. That's it. And for the same set, we take the mouse. Wait a minute. Here we take the human sequence and we put the variant into it. No, wait a minute. What? I'm confused now.
Into the mouse.
Now here we take the ortholog. Here we take the ortholog, and here we take the mouse protein. So. I have to get back to you on Tuesday. Here's another story, and that one is simple and I remember it. So now what we do is we take this position, so we have an OMIM variant, 5,661. And these proteins, what I take is I'm going to change any residue in that position. So I do all 90 non-native, and I ask what's the effect of those. And now I ask, in my protein, I have a set of residues that have a similar level of conservation. Okay? If I pick the one, so the conservation between two proteins, now compared to proteins, the top conservation is different. I don't care about that, I always pick the one with the highest. And it turns out this tends to always be an OMIM variant. So OMIM turns out to be exactly in the position. So again, we have 5,660, that's a subset because of a variety of reasons, but in these many OMIM variants, essentially they always put themselves onto the most sensitive spot in terms of conservation and in terms of sensitivity to variance by this prediction method. That I found, again, is an interesting thing you can do with a tool that has so many limitations. And on top we can go beyond singles, we can sort of milk the methods more, since now we can, in this particular case I show you 3 residues that have a variant known. What we can now do is we can sort of cycle through it and at every single position do 19 non-native. So at every single position we can ask what would be the prediction if I predict all the 19 amino acids that are not observed in that sequence there. And I can do that for every single position in the protein, and that gives me sort of a profile. That gives me a landscape. And one question that I can ask with this landscape is, when we look at the amino acids, we ask, so we replace a particular amino acid against alanine. And that gives an effect. And now we are asking, how does this replacement sort of correlate, and there again is some issue in this story, there's some prediction mistake, but how is it correlated with this substitution? How are these correlated to doing them all? So I do all 19 non-native versus I do only one. So I replace every single position against alanine. So for alanine it doesn't work. But if a residue is not alanine, I replace it against alanine and I measure the effect. And then I do 19 non-native. Okay? So I'm asking here, I do then first alanine, then cysteines, and 19 non-native for every single residue in a protein. And then we do that position against either of those amino acids. We do what people historically call an alanine scan. So we mutate every residue into alanine. And we ask, how does the predicted effect for an alanine scan correlate with the overall average, so 90%. And then for cysteine it's sort of close to 90%. And what you see here is, in fact, you can say that, yeah, well, that is trivial. People expected this. This is why they did alanine scans. Because alanine, thank you, Daniel. Because Paul, you're going to have to decide.
I'm very good.
That's a great idea, but I believe it's also the air story. is warmth and air. So ultimately the point here is that the method picks up that something that people know, that alanine is the best way to sort of scan an average sensitivity to mutation. Machine learning method to learn something about the biology or about the problem underneath. Here's another story.
Oh, it's frozen. When you're using the two things together, you accidentally clicked on the freeze button. Not on the white one, on the overhead projector.
Oh, okay.
On the white remote, you click the freeze button.
What edge?
Now it's working again.
I didn't even know there is a freeze button. How did you know that?
I
saw the freeze thing. Yeah.
No, I saw the freeze thing pop up while you were smashing the remotes together, and I imagined.
Ah! Thank you. So yours. Anyway, so here's P53, and I essentially do something like a difference of the sensitivity of a particular position with respect to everything. So I have a bunch of averages that I compute. That's the residue position. So the protein begins here, ends here. And what you see is there's something that stands out in this protein in this region. And that's exactly where the P53 domain sits. So we suddenly, what we are measuring essentially is we predict the effect of every single position, variant of every single position upon molecular function. And we have some evidence over the protein that tells us there are regions that could be domain-like. Yeah?
What is the negative value here?
So this is a difference from the average, from the overall average. and then there's a log, so there's a little bit of more of a computation. But ultimately, less than expected. My point here is, the signal that I'm plotting has to do with the effect of 19 non-native per residue, plotted out on the protein, and I see a differential behavior. And in this particular case, this is the first sample that Jana checked for her thesis. This was just last minute before the thesis came into her slide, and she immediately saw there is a signal for something completely different in there. She just generically, not for me. Anyway, so another thing that she did, and I'm going to, due to time, go over it quickly. So there's some signal here that has to do with hotspots. So this also carries some signal for hotspots. And here is an example, and I'm sorry for the quality is slightly better on this slide here. Let's just focus here. Let's not discuss what differs here. Actually, it's best here. Mean over non-native. No, it's best here. So there's red. You may see red. So there's a density of red here. A density of red means essentially in this average overall, 19 non-native, what are the most sensitive, the hottest spots in that sense. The red is most sensitive to variation if I do something like 19 non-native. And the differences here are the way you sort of average and what exactly you look at. Now, this one happened, the one that is sort of in my old version there was still a rotation, and you saw that the sort of most red is really around the binding site. And the second most red is in fact around that binding site. It's more difficult to see. But there is a signal, and there's a strong signal. In fact, are we running? Yes. The story that Maria talked about, the prediction of binding sites, small molecule binding sites, that picks up exactly on the signal. Her most important input signal is the sensitivity of that particular residue for change. That is what she uses. Ultimately, it's the most efficient use for input in her method predicting small molecule binding sites. Here's an application to something else, and I'm again, given time, going to zip through it very quickly. The point is, we simply take a particular receptor, melanocortin receptor, relevant for diabetes. These are the 7 transmembrane helices, and we essentially map out what are the most sensitive spots. And then we look at a second receptor that is sort of 60% sequence identical, and we compare them. And in the comparison, we can see positions here, such as the T, where one is red and one is blue. One red and one blue means their sensitivity to change is different. So we have an example of a method that has the same amino acids. So we're looking at two, what we would call homologs, two 60% identical proteins, and we can see that there's a differential behavior at sites where they have the same amino acid. And that's a unique thing. It's absolutely, I mean, any kind of method that looks at functional impact, yes?
Do we know if the folding is the same?
They're both 70M, so they both sit in the membrane. So we don't have structures for them. We would, on the level of structure prediction, we would say we can use one structure to do the model, homology model for the other, and therefore we would argue essentially yes. That's correct. And we would argue that the minute differences that are at the end of the helices, they affect a lot of the binding features that they have. So essentially what molecular biology is in these loops is relevant for how they bind, what they bind. But the unique thing about this method here really is you can look at cases where you have the same amino acid and you still see some differential behavior. And some of that has, in the aftermath of it, been historically verified. Okay, but you have a sort of landscape of mutability. We actually don't quite know all the SAFs, but for instance here in an example of a map, a heat map, where we have the amino acid sequence running here, the protein sequence, red, and these are the 20 amino acids here. Red, totally red means here's the native and black, and every variant or every SAF here against any other amino acid has a strong effect. That's why it's red. And you have a lot of red because this protein really is very sensitive to change, yes?
What does SAV mean?
That is, SAV is SAF, but it's not on this slide.
Yeah, what is
A single amino acid variant. Thanks to Paul, we have SAV is essentially one amino acid changes. Yeah? And so we can use this almost all red map, put it on top of a structure, and make sense out of what are the most likely candidates in terms of their. So we have two places that are red and closed in 3D space. If you have a structure for this particular example, and in this particular case, Max did that and came to a bunch of residues that really relate to the phenotypically relevant binding sites of that protein. So you can come a lot with a map that is so red. So there's a lot of information in this mutability landscape. And now we get into, no, we're not going to get into that because I want to go to another point. So there are now deep mutational scanning studies, and they really do what I said. So they take one particular protein and change essentially every single residue and measure the effect upon molecular function. So we have now a data set of 20 proteins of this type, and we have a difficulty to measure how well methods perform. As simple as that. Why do we have difficulty? Yes?
I just have a general question. So if this worked so well in animals too, and we saw that you could find genes, could you just like throw it onto unknown animal genomes and try to get good indications of where genes might be?
No, and the problem is, again, I'll get back to that in a moment, too many things are predicted to have an effect.
Oh, so it overpredicts. So that was like,
yes, we don't know, but we are now...
That was a nice example a little bit with the...
For some extreme examples we may do that, yes.
Oh, okay, but it's not, you can't do this genome-wide.
Not for that purpose. Again, we can do it genome-wide for some issues. I'm going to present one. Now, even on that level here, we are not sure whether we feel that the methods that we have today really correlate with those data. Those are new data sets. Now we have machine optimized methods that use those data sets, but the traditional ones don't. And whether they work on those or not is a matter for debate. Why I say that is I have a bunch of slides and they give different answers. Ultimately, it's unclear to us what the answer really is. Different answers as in, so one way of, let's go through these very quickly. The underlying problem ultimately is that the experimental data sets differ completely in their scales and what they measure and how they measure. You can have an AUC plot, an AUC number, you can have a ROC curve, and in the ROC curve some methods seem to be better than others. But then you look at the correlation, and the things look different. One thing in this plot here, for instance, by this measure, we see that more recent one that uses these deep scanning data here is seemingly worse than something like SNAP, or than even the simple conservation here. But then when we look at the entire data set, at the raw data, and even that doesn't exist, it's not raw data, some of these scales measure, I don't know, -5 to 50. And on the same protein, on the same sort of token, somebody else measures on a scale of 0 to 1. Now you can try to sort of align these scales, and that's what we do here. But it goes sort of wrong. But anyway, so that's what we do here, and then we can look at one particular method, SIFT. Each dot here is one amino acid change, one SAF, with an experimental measure. And we are asking, is the experimental value that has effect on 1 and almost no effect here on 0? And it's a subset of only the ones that have some effect as opposed to the ones that are beneficial. So this is the experimental data set. Up here you see the distribution of the experimental data set. So what you see is that in the experimental data set, most variants do not have an effect. Okay? And here we see SIFT. We see SIFT, no effect, bless you, strong effect. Here on that axis, you see the distribution of SIFT. It's just the opposite. SIFT predicts that almost everything here has an effect, so it's completely wrong on the level of the distribution. This is not on an individual level, although you see that some clearly are on the diagonal, so some are right. But overall, the distributions, the tendency, so in that protein, SIF would say most variants have an effect, and the experimental data here would say the opposite, okay? Now this is for polyphen, again, sort of the same overall trend, although you would argue this signal clearly in terms of linear fit, this looks better than this. And then we go to this method here, Envision, that is the one that is sort of optimized for the data set, and in fact that has the lowest Spearman of all the 4, yes?
I was just about to ask what the x-axis means because I didn't understand.
So this is the experimental measurement. again, is a normalized experimental measurement. So there is an experimental measurement of an effect of a particular variant upon function. We do that for 20 proteins. The whole data set, I can't remember, is maybe 40,000 residues. So there are many variants that are tested here. So this data set is larger than what I showed you before for the machine learning. So it's a fairly large data set. For, however, a very tiny subset of proteins, 20, or less than 20, fewer than 20. And this is effect measured on the one, and 0 is no effect measured. The Y is 4 different prediction methods, again normalized to the same value. I told you snap values go from minus 100 to plus 100. It's just normalized here. Okay? And now what you see is, again, this has the lowest Spearman, but it's the only one that captures the signal right. So this is the only method that would say, well, most variants, at least in this particular set here, appear to be not having an effect. That's an important signal to capture, right? So Spearman is also not completely telling the story. And we cut this in so many ways, and every single time we have a new measure, we get a different answer. And that ultimately is, and then finally we decided that we want to send it because we get some averages, and the averages we see some trends, and then we publish these averages, and then we say afterwards it's not meaningful because although seeing this overall trend, we actually don't quite understand how to slice the cake. And then there are other publications that sort of see a similar signal. And I'm going to click over this one here. So this story is about sort of doing the same thing that I talked about now, so effect, but I'm no longer defining effect as effect on function. I'm defining effect as effect on protein structure, and develop a set of methods that do that. I'm skipping over it. Now I'm getting to something else. I'm getting to this old idea of what now is called precision medicine. Which is ultimately the question, how, if we now know for all of you, you can get your genomes from Nebula or from 23andMe, and what is the fact that you have your genome going to be the effect upon seeing the physician? How is that going to affect your treatment? Well, one way is sort of we have for each genotype a different, let's call it treatment. In order to do that, there's a bunch of things we have to do, set a bunch of methods right. We know the human sequencing at this point. We know there are 3G letters fit into these 4,000 books. They roughly correspond to 20,000 proteins. And we actually don't quite know how many genes or proteins we have. But let's just say we have 20,000 proteins. We know there's diversity. And the first question is, how much do we differ? In terms of, I'm not asking for what historically, so we zip the word SNP. SNP is no longer arms. The blue ones, you and your neighbor have the same amino acid. Red ones, you differ. Well, what does it translate to? Actually 20,000. So in every single protein you have a difference. On average, right? That's one way of thinking about it. Now this, you may say, is not a lot, but don't forget this entire library, we are talking about germline. The genome is the same for my 37 trillion cells. The library is the same. So now the question becomes, do they have an effect? How would you check?
Only looking at proteins, so this is completely regulation. And
forget about regulation.
Okay.
Only proteins, yes. True. Which, mind you, again, for most rare diseases, by far most rare diseases, in fact, hit proteins.
Yeah.
So I'm not saying that regulation is...
Or the easy ones that we can see.
Well, I don't know if you sort of postulated if we had another 20 years to go, do you believe that most would be outside of the protein?
I don't think most, but I think there would definitely be a higher percentage.
Than today, yes. You're totally right. But I'm not entirely sure how much. I have no idea where the ratio will go. Anyway, let's get back to this question here. We have the 20,000 amino acid changes. My question is, do they matter or not? What would you do?
Run SNAP.
Well, is that the first thing you would do? First you would not have snap, right? Or, well, I mean, what most people would do is essentially they would look up experimental information. For how many of these do I know? I have already 5,661 in terms of OMIM. So let's just look up all of these. Turns out this entire set, don't forget we have 100 million possible variants in a human genome. And this 561 is a tiny number compared to 100 million. It turns out that whatever we have experimental data for is less than 0.1 per mil of this. Since we are on the 0.1 per mil, at least for some of the people in this room, SARFs in them. And for I mean, essentially with experimental data we don't get there. Here I'm writing maybe in 2050 we get there. I'm not even sure about that. And meanwhile, Paul is totally right. What we can use is prediction methods. That goes back to Jana, who sort of developed one, the latest one from Max, the latest improvement from Max, still with Jana. Now the point is we have effect and neutral, and for us effect could be two different things. Yes?
And just one question about these 0.1 mil. Do they also include variants where we did not observe a disease?
So yes, so for this 0.1 per mil, what I'm looking at is I take every single variant for which I know what it does somehow. I have some characterization of what it does, effect or not. Then I look at all the proteins that have a SAF, and I'm asking, does it have a binding site that is related to a position where I don't know whether that variant is going to, but I know the variant is in a binding site. Then I can say something. That is all included in there. So it's not really specific for that variant. Then the number would be even smaller. So again, my point is that effect for us is it doesn't function at all, or it has a new function. We cannot distinguish. It's just a radical change. And we have cases where SNAP has a very strong score for something that is in fact a beneficial change. We just are blind on that. Again, the SNAP score is between 100, strongly predicted effect, minus 100, strongly predicted to be neutral. If
it can find benefit, if it can't distinguish between beneficial and harmful, how does that work with the evolutionary information?
It doesn't. I mean, that's one of the big issues. But the part that I suppressed by looking at the DMS data, no method currently really reflects beneficial variance.
Yeah, but if you're basing a lot of your prediction on evolution, and all the beneficial ones would be actually allowed and even wanted by evolutionary data, so how can you then?
We actually don't quite know that, Paul. So we call an enzyme to gain function if it does something that it couldn't do before, which is not necessarily what will help the enzymes in the body. So ultimately we don't know what the body optimizes. It may not be to have an optimal binding constant or something like that. All of the kinds of things we do, particularly in industrial purposes, that we have an enzyme that sort of can withstand high temperatures, is not what evolution optimizes for. Evolution has sensitivity to change as something, or stability ultimately, right? A different stability, not the binding stability.
Yeah,
yes.
in general.
Stability against change, or stability for doing the body job. Anyway, so getting back, say I had a data set of amino acid of changes that I know experimentally have an effect, and I had a method that is very good. What I'm looking at here is cumulative numbers. By cumulative numbers, so I'm asking for all in my data set, for all the variants where the prediction score is above 50, what is the predicted effect? Or how many of these are predicted to have an effect above 50? So in this particular case, the answer would be sort of close to, let me just go to the next animation. Let's pick this point here. Above 82, 98% are predicted to have an effect. Okay? So if I had a very good method, then it would predict something like that for the ones that have an effect. And most of the ones that are neutral are predicted with a very, very strong neutral score. This would be a good method. In reality, my method is this. And that's the cross-validation reality. So that's experimental effect and experimental neutral. Again, it's a data set of these 40,000 that I showed you on Tuesday, where we have experimental data mostly for molecular function. There are some of those that are impacts on the system, but most of those are molecular function. And this is what we have. Now, if you sort of look at it, this part, the degree to which this is not up here is the mistake, or the degree to which the green one is not down here is the mistake. And conversely, the degree to which green and red are different is the power. If green would be the same as red, the method could not distinguish. It would say always give, on average, the same answer for a variant with an effect and not, right? That would be a total random method. So essentially you would argue, it seems that the method is much less good, so there's sort of less power in putting them apart than what you lose with respect to, there's much more you can still go on this plot, right? But this is what I believe is the best what we have. And again, this method has been optimized on SAFs for molecular function. Now what would happen if we apply OMIM? So things where the effect is on the molecular, on the system. And that's the orange curve. So here 2 points. The first, I have to repeat, or not repeat, I have to make that clear. None of these orange ones here was used for training. Okay? None of them, 0. So there's 0, so we use a model here where we have essentially we retrain and then kick them all out. So make sure that 0 has been used in this particular case. Is there anything that jumps to your attention? Is there anything else? So, where's the curve going to fall? We're talking about people who are healthy. So this is done, my definition of healthy is they all could be sequenced. So none of the people who have been sequenced died before they were born. They all could walk to somewhere, so that those are all ways of defining healthy people for me. So none of them suffers of a major disease that affects their life in a way in which it prevents data collection. Let's put it like that. That's one definition of it.
The orange.
No, now I'm talking, no, they in fact, many of those will not go. They go to they're being sequenced because they're diagnosed with a disease and then the doctor sequences them. But now I'm talking about the 20,000 between you and your neighbor. Where will they fall? So I'm going to show you on the next slide the result of predicting for the differences between you and your neighbor. Where will they fall? And that's now what I'm hoping for is a prediction from you. Yes, Yannick?
Well, probably on non-conserved amino acids.
Yes, no, I mean the curve. So I'm going to show, if I ask you to draw a new curve here, where would you draw that? And that curve will reflect the effect of the 20,000 between you and your neighbor.
Probably near the green.
That's what we expected. And of course I show you this story only because it's not the expected. And in fact, because it was not the expected, it's a new discovery. Because it's a new discovery, it's extremely difficult to get this out. Took years, and many students to ascertain this. Took years to publish this. The quote is actually missing. It was published 2 years ago in Scientific Reports. Simply because nobody could sort of believe this. Yes?
Are there people in these groups from different ethnic backgrounds?
Yes. I will not quite get to this point, but I will get to some aspect of that point in a minute. Let's first go down a different route. The first thing that I want to point out to you, sorry, I didn't realize it's an animation, is that 1/4 of the data set has what we would call a fairly strong effect. So 5,000 in your 25,000, in every one between you and your neighbor, what we would predict have a strong impact on molecular function. So this is way more than we thought. And this, either we are wrong or it really is big. Next question that we had, well, what if we compared ourselves to a different species? Let's look at a hominoid. And ultimately this, why I'm asking this is your idea may be, well, sure, my neighbor looks different from me. So I need a lot of variance, right? That means when we compare it to Neanderthal, we will see even more difference, because most likely your neighbor is more similar to you than Neanderthal would have been. Yes?
So what I don't understand about drawing this purple line is, so there's two things. There's one's like the difference between me and my neighbor,
yes.
And then there's the difference between a mutation that has an effect versus one that doesn't have one.
No, this is only, the purple line is only you and your neighbor, right? Mm-hmm. And what I'm saying is that if I sort of look 25% of this variance between you and your neighbor, translating to 5,000 for you, 5,000 of the 20,000 variance to your neighbor, have an effect above 50. Predicted effect above 50. For us, an effect above 50 is a very strong effect upon molecular function.
So 25%.
Effect of 40.
Of all the variance between you and your neighbor.
Stronger.
Yeah.
Take the variance with the highest effect, and that's the bottom right. And you take the one with the next highest effect, and that's you plot your points.
But like, if neither none or my neighbor have a disease, what does it mean?
Right. That's exactly the big surprise. Yes. How come we have so big differences? Yes. Exactly. So now my next question is, what if we compared ourselves to Neanderthal?
Exactly the same.
Denisovan. So Denisovan is here. It's so choppy, this curve here, because we have one individual at this point. Now we are at this point. So at the point of this slide we had one individual. Now there are more. And this is going back to Svante Pabo and Janet Kelso's work. Let's go one further. Gorilla. We at least have a population. Now we sort of turn this machine learning the other way around. Now we have sort of a neutral data set. Now we redefine this as neutral, have a neutral data set that works better than my training set. And in fact, since we discovered this, we sort of have used this for training for some of the models. And it goes on for mouse. Now, clearly, some of those matter a lot, and our genetic, some of those are like disease variants, and some matter. But what we would argue now is those that matter may be rare. So rare, what I define as rare on this plot here, what we expect is that the rare being less than 1% in the population, as opposed to frequent, more than 5% of the population. And there we get into this issue of subgroups, right? So essentially you could argue these are outliers. These are variants that just a few individuals carry, and because just a few individuals carry, it seems that they're evolutionarily not getting through. While the ones that you see in at least 5% of the population, they sort of at least are accepted in some group locally, overall. Okay, so some of these data sets, so the next slide is going to be, or the over next slide is going to be on 60,000 people, 10 million SAFs. So this includes a lot of subpopulations, includes a lot of populations. It's a large, relatively large data set, all right? So what we expect again is that rare, just, there are random mistakes, so to speak. Some, but they don't survive, and that the frequent ones have more of an effect. And everybody would sort of expect that. And again, I'm showing you because of course that is nonsense. The real result is the other way around. The common in green is above the rare in red. Forget about the orange, it's just in between. But clearly, again, the error of these curves, since we have 10 million SAFs in here, the error of the curve essentially is not distinguishable from the curve itself. So these clients clearly differ. What does that mean? We actually ultimately, and we're going to leave you alone with this, we actually don't know. So in a paper that Jana Bromberg published, she said that ultimately you have a thing where weak effects sort of add up and define your individuality. So it's a little bit of an effect, not a big effect, a little bit of an effect that sort of adds up to becoming who you are and how you differ. The larger differences are subpopulations have a different way of scenting, or we know it for our scent receptors, for instance. They differ in subpopulations. And those kind of things, maybe those are some of the things we see. Or maybe to some extent we have this example here, where we know back to the sickle cell anemia. The sickle cell anemia is a bad mutation, bad as in causes a disease, that has a positive side effect. If you get malaria too, this helps you to increase the resistance against malaria. It's the same variant. It's a single amino acid change that causes two things, resistance or more resistance against malaria and sickle cell anemia. One is bad, one is good. And maybe some of these variants that we actually have in the population really are there because they could help in some circumstances. Anyway, that is the malaria story. And maybe we have in all of those that have some effect, maybe some of them are good. They make out help me to look different or be different or help the population, and some of them are bad. And bad and good could be for me or for the population, right? There are different levels of defining bad and good. Now, maybe we could sort of find a way to sort of damn the bad effect ones, attack the bad effect ones by, for instance, personalized medicine or precision medicine. So essentially by sort of looking at something where my genotype is specific to the drug I take. Could you imagine a situation in which each of you gets a genotype-specific drug? Sir, for me, when I first saw this slide, now it's a few years ago, but it's actually not that different. So that's FDA only, US, but essentially it's the same worldwide. The European Union has a similar agency. So a few handfuls, 40 in some years, it's 15. Somebody said 15, so there are years with 15. And this again is new drugs. So it's not like the same chemical ingredients and call it a different name. So these are really new drugs. It's going up because we are also targeting things that are less trivial. We are targeting higher hanging fruits. And the story is getting increasingly difficult. Ultimately, this is also leading to a merger of more and more pharmaceutical companies. There's a variety of results of that. Ultimately, my message is, it will not be a genome-specific medicine. It will be a genome-specific, a genotype-specific diagnosis, choice of drugs and food, lifestyle, ultimately. We will choose and that happens already today. So you all talked about in cancer, particular cancer, there are choices of the type of cocktail you get. That is genome specific. That happens already, right? And those are the kinds of things that happen already today, and that will happen more and more. We will not really develop a completely new drug for that new genotype. We will sort of combine things in a different way, and we will eat differently. And we'll live differently, and ultimately this will help us to somehow reduce some of these bad effects and sort of focus more on the good effects. Now for me, I have two flip sides of the coin. There are a lot of variants and they matter, and they are good for the survival of the species, but not possibly good for my happiness. So I don't like them. I mean, it's good that the species survive. I'm really happy about that. But I also want to be happy for myself. And somehow precision medicine or these developments help me to hope that the probability that I also benefit will increase. So somehow that means that I will ultimately check some of these, and I sort of will combine the best of both worlds. I will gain from diversity, because that is exactly why evolution tries out these changes, because we need diversity in a group. So I will benefit from diversity and still be happy. And that for me is a sort of great outcoming. I will keep it here.